Yay. Okay. Get the code if you still want the code. Hi, I am Mrs. Sloan, and this is my honors bio 7th period, who is awesome. Say hi. Oh, that was so sad. There actually is Friday. Okay, so we'll go ahead and start. This is our chapter 23 review, Patterns of Inheritance. And I'm going to really focus on the conceptual issues. We got a lot of practice last class where we did a lot of practice problems. And I have linked that presentation in here if you want to have access to it. Besides, it's posted on Google Classroom. Besides, it got emailed to you. But want to make sure that you can differentiate between Mendel's 3 laws. Can you provide ratios, genotype and phenotype for crosses, whether they're monohybrid or dihybrid or trihybrid? Can you identify exceptions to Mendel's law? Like, do you know the difference between incomplete and codominance, multiple alleles and polygenic traits, and linked genes? And then environmental effects on genotypic expression. Can you identify if a pedigree could be autosomal dominant, autosomal recessive, or sex linked? And have you done your additional objectives? Okay. Tell your bio buddy on a scale of 1 to 10, how are you feeling about this right now, with 10 being awesome sauce? Okay. So the father of genetics, you want to remember Mendel. Hi, guys. Father of genetics is who? Mendel. And what did he work on? Peas, garden peas. Yeah, because he could cross-pollinate them. They had fast reproductive times. He could get thousands of offspring. He was really good at collecting data and using statistics to analyze that data. Did he die knowing of his value, his contributions to science? Mm-hmm. Yeah, he died before knowing that, right? It wasn't discovered until about 50 years later. And he used a lot of really obvious traits that had clear dominance, where it's like tall over short, round over wrinkle, right? Yellow over green, all those things. So when we talk about these alleles, when you do all your Punnett squares, right? Whenever you do a Punnett square, okay, there we go. What you're doing is you're putting one parent's alleles on the edge and then the other parent's alleles on the other edge. Those are the possible gametes that they could have provided. And we have to take into consideration when we put those gametes, if it's big T, little t, we have to put both possibilities down, both the big T possibility and the little t possibility. So that's why we list them both. But what law says you will never give both alleles in your gamete? Law of segregation. When does the law of segregation occur? Anaphase 1. Anaphase 1. What law says that if you have a combination of a big T, little t in a zygote, because these are the zygotes right here. I don't know why my board is so slow right now. These are the zygotes right here. What law says that this plant right here will be tall? Law of dominance. Can I see the law of independent assortment here? No. When I am looking at alleles on the same chromosome, they do not assort independently of each other. What do you call those genes that are on the same chromosome? These genes are all what? Linked genes, right? Wherever big R goes, who's going with it? Big S, little t, and big G. They are linked genes. They travel together. So you will not see independent assortment amongst those. What's the only thing that can break up gene linkage? Crossing over can break up gene linkage. Okay, ask your bio buddy. You got that? Are you feeling strong on that? Now, I'm going to take one pair of alleles here, big R, little r. Okay, and what if there was another set of alleles, this, on a totally different homologous pair, right? Where one was a big T and one was a little t. In metaphase 1, could they assort independently of each other? So I could get the big R with the big T, or I could get the big R with the little t. So that's why when I'm writing those gametes, I write big R, big T, big R, little t. But I could also get the little r with the big T, or I could get this little r with a little t. That's independent assortment. Okay, is that like clear in your head? Okay, questions you want to ask me about that? All right. So now when we look, sorry, I've got to erase a little bit here. Oh, that's a monohybrid cross. You know what that is, right? You want to see it again? Mono means one. Hybrid, right, means that this is a hybrid. What's another name for that? Heterozygote, right? This is heterozygote. What would you call this? Big T, big T. How would you describe that? It's genotype. Homozygous dominant. What would this genotype be right here? Homozygous recessive, and this is heterozygous. Its phenotype, what it looks like, what would this plant look like? Tall, and this one right here would also be tall, and this one would be tall, and only this one would be what? Short. So you have two different genotypes for the same phenotype, right? So that is with clear dominance. Big T is dominant over little t. Is it always like that? Give me an example when it's not like that. What is it called? Codominance is one way. What's another way? Incomplete dominance. Exactly. That's another way. Or when you have a whole bunch of different alleles contributing, like your intelligence, what is it called when you have a whole bunch of different combinations of alleles, T's and R's and P's and Q's and L's and M's all working towards your intelligence? Polygenic. Multiple alleles would be this, just because you said it, so I want to differentiate between it for you. Multiple alleles would say you don't just have big T and little t. You could have T1, you could have T2, you could have big T3, and then little t. That's multiple alleles. There's still only two spots right on your homologous pair, but you have more than just big T, little t. You have multiple alleles. So you could get T1, 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 T2, T1, T3. Are you with me on this? T3, little t. That gives you a lot more genotypes and more than likely more phenotypes as well. Whereas polygenic, this is multiple alleles. Polygenic means many what? Genes. So a polygenic trait could be something like you have big R, big S, little s, anytime. As soon as I write again, right, it's going to do it. Big S, little s. You could have T1, T2. You could have big IA, little I. You see anything? All of these letters working collectively for your trait, that's polygenic. When you have options on the T besides big and little, that's multiple alleles. Questions? Ask your bio buddy. You okay, bio buddy? If you don't know your new bio buddy in 7th period, introduce yourself. Tell them your name, even if you do know them. Maybe they don't know your name. Okay. Now, law of dominance. Where in here do I see the law of dominance? Where specifically do I see the law of dominance? Tell me out loud. Big Y, little y, right? In the heterozygote condition. Here, of course, I'm yellow. Here, of course, I'm green. But in the heterozygote, that's where I see the law of dominance, because big Y is dominant over little y. Perfect. Okay. Law of segregation. These are alleles. Those alleles. Now, you know, during the S stage of interphase, you will make sister chromatids, right? That'll be exactly like that. But the homologous pairs separate in anaphase 1, and that is the law of segregation. Big T gets separated from little t. But if this is big T, both of them are big T, right? Those sister chromatids are going to separate in anaphase 2, And these two little T sister chromatids are going to separate also in anaphase 2. But the law of segregation is when you separated the alleles from each other. Okay? And then the third law, oh, are there exceptions to the law of segregation where it turns out normal? No. Are there exceptions to the law of dominance? Yes. Are there exceptions to the law of independent assortment? Yes. Linked genes, like I showed you, that's the exception to the law of independent assortment. And what breaks up linked genes? Crossing over. Good. All right. And then here I'm showing you the law of independent assortment. This homologous pair could have lined up this way or it could have been flipped. Each homologous pair can line up any way it wants to relative to the other homologous pair. You don't have to have the dad chromosomes on the left and the mom chromosomes on the right like this one, right? And how many homologous pairs are we dealing with? 23 homologous pairs, because how many chromosomes do we have? 46, right? So all 23 can line up any which way they want to. All right, and that's why we get, when we look at this genotype, we get, that's the genotype. These are the gametes that could come, and this is little b, little e here on the bottom. These are the different gametes we could get from it. All right, anything you want to ask me about that? Okay, so in this one slide right here, you can see all three laws here. So I can see the law of dominance whenever I have to look at the zygotes to see the law of dominance. I have to look at the zygotes because that's the combination of alleles where one allele may be dominant on the other. That's the place where you have both alleles present, is in the zygotes. And any heterozygote in here where it's big Y, big R, little r, for instance, it's still yellow and it's still round, even though it's carrying wrinkledness and carrying the green color. Okay, law of segregation, I see that when I look at the list of gametes, because I can see I have a big Y with a big R, but I also have a little y with a big R. That's the law of segregation. They can separate independently of each other. That tells me, because I'm seeing the law of independent assortment there. Oh, sorry, I don't know what I just said there, but can I go back to law of independent assortment? Sorry. Because I'm seeing a big Y with a big R and a little y with a big R, I know that the Y and the R are on two different sets of homologous pairs. Does that make sense to you? And so as a result, that's why I can see all the different combinations. The law of segregation is because none of these gametes have two Y's in them or two R's. Why? They were separated from each other. Okay, questions you want to ask me about this one? Okay. All right. Next. Okay. I wanted to use this slide to remind you that chance has no memory. So the first son I had was a boy. Does that mean the second child I had, or the first son I had was a boy? Good, Mrs. Sloan, biology. The first child I had was a boy. That was Caleb. Okay. My next child, did it have to be a girl? No, because chance has no memory. I had another boy. My third child, did that have to be a girl? No, chance has no memory. So you want to remember that. It's not going to turn out every single time, whatever the odds are, they're repeated at each individual event. Okay, and next. Yay. When would you need to do a test cross? Do you remember? Yes. Right. So you're expressing the dominant phenotype, and you could be hiding a recessive allele, right? That's when you don't know the genotype. Exactly. You could be carrying, if you had brown eyes, you could be big B brown, or you could be what? Big B, little b brown. So who would we cross you with if you had brown eyes and we were trying to find out your genotype and we didn't know who your parents or your grandparents were? Who would we cross you with? Somebody who had blue eyes. Because we know they're always going to give the recessive allele, and so we look to see what your zygotes contain. So in this case, we didn't know if this purple flower was homozygous purple or heterozygous purple. So we crossed it with the recessive trait white. And if every offspring flower turned out purple, then it was more than likely homozygous purple, right? But if just one, only one had to come out white, you know it was a heterozygote. Okay, you good? Okay. So now, beyond dominant and recessive alleles, these are the exceptions to Mendel. So the first exception, hang on, my computer's wanting to update. No, do not start now. Hang on. Meh, meh, no. No. Oh. Oh, it's not letting me. Okay, come here. Start in 4 hours. Submit. Okay, that would have been sad. Okay, so this one, blood type, is great because there's two things you can see here: codominance and multiple alleles. Multiple alleles, because I just don't have a big I and a little I. I have big I superscript A, I have big I superscript B, and I have little I, right? That's multiple alleles. On top of that, both the big IA and big IB are both equally dominant, so we call that codominance, like co-partners. Both of them get expressed. All right, but any big I is still dominant over a little I. Yep. So here, if I said cross a heterozygote A, that's a big I, a little I, that's a heterozygote A, with a heterozygote B, big I, B, little I, then what are the potential offspring? This right here is a genotype. What would be, this is the genotype. What would the phenotype be? What is it? AB blood, exactly. What about this one? B, and this one? A, and this one? Oh, good. Those are the phenotypes. The genotypes are listed right there. Check with your bio buddies, see if they're okay with that. All right. Next, incomplete dominance. It's different from codominance because incomplete, like neither one really wins. It looks like an in-betweener, right? This is your black rat crossed with your white rat, and all the hairs are gray. Red flower crossed with white flower, and all the babies are pink, right? Baby flowers. This is, when you see here, straight hair and curly hair. Straight hair is H1H1. Curly hair is H2H2. When you have your zygote H1H2, you have just what kind of hair? Wavy, neither straight nor curly. Can two wavy-haired people have a straight-haired child? Two wavy-haired people, two of them have a straight-haired child. Yes, what are the odds? 1/4. How about two wavies having wavies? Half. How about two wavies having curly? 1/4. Got that? Each genotype gives its own unique phenotype. Okay? And then, yeah, let's get on that. Are you all right? So incomplete codominance, right? The example I always use is that rat. If it was codominance, a black rat with a white rat, you would have both black hairs and white hairs. It might look gray to you, but you have both colors. But incomplete dominance, all the hairs are gray. Incomplete code, you have two. Okay. All right. Next, sex-linked inheritance. For sex-linked inheritance, what I want you to focus on is the X in sex. Okay? Because I'm not going. Okay. Good. Good. Okay. Sorry. Do you know who that is? He's a math teacher. Okay. So in sex-linked inheritance, the X would be found on the female, yes? Can you find an X on a male? Yes. What? Remember, male are XY, females are XX, right? So females have two options on their X. So if we get one bad X, hopefully we still got a good one, right? Guys have one option on their X. If it's bad, it's bad. If it's good, it's good. What are they not that girls can be? They are never carriers. They can't hide that trait. They've got it or they don't have that trait. And all the sex-linked crosses that I am going to give you, they are always recessive. So they're going to behave like a recessive gene, right? And this is your exception, another exception besides codominance and incomplete dominance. Sex-linked inheritance in boys is the exception. Because normally to express a recessive trait, you need how many? 2. Boys only need 1 because that's all they got. But normally you need 2 to express a recessive trait. Boys only need 1 because that's all they got. All right, and then polygenic inheritance. We talked about this a little bit already. Polygenic, poly genes, many genes. So you have both A's and B's to give you your skin color. The extremes would be homozygous recessive and homozygous dominant, right? And you tend to see a bell-shaped curve. It's not tall or short or ABO blood. You have 1, 2, 3, 4, 5, 6, 7 different phenotypes when you have polygenic traits. So that's how you would know. If you have a whole bunch of different phenotypes, it's probably a polygenic trait. You okay? All right. And then your environment. And the classic one is the bunny. Normally, a bunny's body would be solid white because it's warm by the bunny's warm body. But if you shave the bunny's back, a Himalayan rabbit, and put a cold pack on it, the environment changes gene expression. The gene says be white, but only be white when it is warm. If it's cold, it's going to be what? Black. So your environment is influencing your phenotype, what you actually get from your genotype. Okay? So, for instance, if you had genes to be really tall, but you were malnourished, would you be tall? No, because you were malnourished. You could be super bright and intelligent little baby, but if your mom was drinking the alcohol or doing drugs while she was pregnant with you, is that going to influence you? Yes. Your environment is affecting that. Okay, next. I already talked to you about this. Linked genes. These are genes that are on the same chromosome, so they're going to travel together. What law do they fail to observe? Independent assortment. What's the only thing that can break up the linkage? Crossing over, which occurs in prophase 1. Okay, and then the last thing we did was we had a bunch of, remember those pedigrees? Make sure you understand how to read a pedigree and what each genotype is. And remember, you got the Google machine. You could literally Google pedigrees with answers, pedigree questions with answers, and you could get more practice, right? Okay, but here are the answers to those ones we did in class. Here on this last page, I encourage you to try the practice Punnett problems that I shared with you. You can do them as many times as you want. You get scored. And also, this is a link to the pear deck from last class where we had practice problems embedded. Okay, you good? I'm proud of you on a Friday, my quiet children that I love. Good job. Good job.